All right, back for a video on neuronal pools. And what we mean by neuronal pool is patterns of neuron connections. What we've seen so far, what's commonly sort of displayed in textbooks and during lecture, is the easiest patterns, which is usually one to one to one. That's called serial processing. And so what we'll do is sort of show you that that's not the only example of how neurons can be connected. All right, so let's start with, and I'm going to use my typical symbol for a neuron, which is just a little blob for the cell body. Again, if it's a multipolar neuron, and I'm drawing these as all as multipolar neurons, which of course is not the case. But it would have dendrites on it if it was a multipolar neuron, right? The axon, and then the synapse, right? And again, we'll see that some of these will have to split to form the neuronal pools that we're talking about. So in the first one, the first type of neuronal pools called divergence. And in divergence, well, the neurons diverge. The information they carry diverges, meaning it splits and goes to multiple targets. And so in this scenario, one neuron receives information, right? So just a little squiggly arrow here, right? And it would then send this information to more than one target. And those target cells would split and send that information, right? And I'm sort of showing sort of bare minimum of divergence here, right? And then you'd have target cells here. And possibly so on and so on and so on. And you can imagine that if we go through multiple cycles of this, we've sort of diverged our information to multiple, many targets. This is really common in sensory function, where information is brought into the central nervous system, and then it diverges to go to multiple locations and possibly generate a multitude of different actions. An example would be you sort of putting your hand on something hot or touching something sharp. And you're going to do several things, right? Let's use an example of you step on something sharp, maybe even easier. Right? So you're going to hop off of that sharp thing, right? You're also probably going to yell out in pain, right? So sort of the reflex to pull your foot off of it would be one pathway. Recognition of that it hurts, right, is a central nervous system function, a brain function, and so that would be a separate pathway. Yelling out that it, saying ouch maybe, would be another pathway generated. Stiffening your opposite leg, right, to make sure you don't fall down when you hop off of this, right? There's another neural pathway. And so we have to sort of take that information, diverge it to a multitude of different sort of processing centers, which then may all generate their own motor outputs in response, right? So again, divergence. Let's go to convergence. Convergence, I think we've seen before because we've talked about EPSPs and IPSPs, and they're a very common example of converging information. Right? And if you think of the term convergence, it means for things to come together, right? And so in convergence, we have something that looks like I'll use different colors here. Right? Like so, right? The information coming from these two neurons is converging on this one. This is a very simple example of convergence. So when we talked about the example I think I gave in class, although let's give it again here, you have control of your respiratory muscles, right? If you want to stop breathing, hold your breath, you can. Yet normally you breathe without having to think about it. And so those muscles are under both conscious and unconscious control. Well, think of those as the conscious and unconscious control pathways. Normally we're not consciously controlling our muscles, so this would be inactive. Right? And our unconscious pathway is sort of controlling it, right? Through stimulatory EPSP generating pathways. Now, if we want to control those respiratory muscles and either breathe faster or slower, right? And we could actually have a third neuron here for stimulatory input. But if we wanted to hold our breaths, right? We could have inhibitory input through a pathway. And then that doesn't mean the unconscious control from the respiratory centers has gone away. It just means it's losing to the conscious control, at least temporarily. And that information is being summated on this downstream postsynaptic neuron, right? So the information is converging, and it can be stimulatory and inhibitory, but it could also be stimulatory and stimulatory in this example on that postsynaptic neuron, right? That's converging information. We'll get to a really good example of this in vision. When we talk about sensory function, get to vision. In the peripheral retina, the amount of convergence is a photoreceptor called a rod, and the amount of convergence is enormous. Thousands of cells converge to single cells, converging information in the retina to sort of like coalesce the input, light input from our environment. Now, it doesn't do that in all regions. It does that in the peripheral retina, not in the central retina. And we'll make sense of that when we get to understanding vision. All right, but anyway, that idea of information coming together onto a smaller number of target neurons is convergence. Now, let's do serial processing and parallel processing maybe at the same time. And we can do those together because it sort of makes sense, right? If we're serially processing something, that's kind of like the reflex arc that we've drawn before, right? Where a sensory neuron conveys information to an interneuron. Now I'm going to have my interneuron split here, right? And that interneuron conveys information to a motor neuron and so forth. So serial processing just means one after another, right? Now, the reason I drew this sort of split, this branch is called a collateral, axon collateral, is that parallel processing is when we then sort of send information along a parallel pathway. And then so in other words, two serially processing pathways running side by side is parallel processing. Now, it doesn't always have to be carrying the exact same information. That gets very complicated. A good example, though, is in vision, where all of the aspects of what we see are not carried in a single serially processed pathway. In other words, light intensity could be carried along one pathway, color could be carried along another pathway, and so forth. Motion along another pathway, and these are very complex pathways. But that's the idea of one after another being serial, and then when information runs side by side, that's called parallel processing. Okay, let's do what's called recurrent. And recurrent processing or connections can be either stimulatory or inhibitory. So I want to use the terms recurrent stimulation or excitation and recurrent inhibition. And recurrent actually means sort of feeding back on. Okay. Inhibition and excitation. I'll draw a single picture here, but we can put a plus or a minus on it. So what we have is one neuron connecting to another neuron, right? And off it goes to a target, maybe. But the second neuron feeds back recurs back onto the first neuron. And again, that can be plus or minus, right? Stimulatory or inhibitory, plus or minus, right? And so in recurrent inhibition, what we're trying to do is shut off this pathway. In other words, activation of this pathway, stimulation of this neuron stimulates the next neuron, but that second neuron feeds back to shut it off, right? So that's a pathway that tends to turn itself off. There are cells that are essentially interneurons that are essentially adapted to do exactly this in the spinal cord, controlling skeletal muscle function. It's one of those, like, we want our muscles to contract when we tell them to contract and not otherwise. And so there are these cells called Renshaw cells that their job is to feed back and sort of say, are you sure? Are you sure? We're going to shut this, motor pathway off if you don't continue to send stimulatory information to this first neuron in the pathway. Okay, so that would be inhibition. Excitation, recurrent excitation, would be when this feeds back and keeps this pathway active. That's sometimes called neural reverberation because it sort of keeps this pathway active for prolonged periods. Consciousness is thought to involve recurrent pathways. There are synchronously active pathways in things like breathing, the respiratory control centers, where we've got these patterns of these cells that are connected that sort of fire repeatedly over and over again, and we can sort of speed them up and slow them down to adjust their collective rate, but we want them to be active continuously. So again, it's a way to sort of keep sort of a positive feedback path loop, right, as opposed to the negative feedback loop with inhibition that keeps this pathway active. Okay, so that's recurrent inhibition and excitation. The last one I want to talk about is called lateral inhibition. Squeeze it over here in the corner. This is really common in sensory function. And what it means is that when a cell becomes activated, it essentially diminishes the activity of the cells around it. And what that does is it enhances contrast, right? And I'll explain that in just a second here. Let me draw it for you. So if we have a cell here, and I'm going to have a stimulus arrive, put it in red. All right, so there's a stimulus arriving. I know, sorry, I can't write huge without running out of space. So we have a stimulus arrive on this cell. It's going to send information to its target cell, right? And that will keep going. But it's also, let's say there's another cell over here that goes to its target cell. And there's another cell over here. This is really common in vision, where you would have a photoreceptive cell next to a photoreceptive cell next to a photoreceptive cell. It's also common in tactile function in skin, right, where we have sensory receptors lined up one after the other across the surface of your skin. And so if a stimulus shows up at one spot, yes, we wanted to activate this pathway so we can detect that stimulus, right? So a fly lands on your arm, right? We want to be able to detect that stimulus. But we want to know where that fly is very, very precisely, so we can locate it on our skin. We're not smacking the wrong spot, right? And so one of the ways that happens is that this guy actually innervates its neighbors. And that's true in reverse too, but I'm not going to show all those sort of crossover pathways, right? And while it was, sorry, I got to squeeze this in. While this guy is activated, I'm going to put a plus sign here. Guess what? This is inhibited, right? Negative sign. And so what happens is the output here is plus, 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 right, on this pathway. And here it's minus and minus, right? Those two pathways are shut down. Even though the stimulus might, be spread out when you depress your skin, it might activate more than one receptor, but it activates one group more so than others. So it activates this group more than others. But by having this lateral inhibition, shutting off the receptors, and you can think of this three-dimensionally as a circle around this too, shutting off the pathways around the focal point of that stimulus allows us to generate contrast. It tells us exactly where that stimulus is. Rather than everybody being stimulated, now it feels like the stimulus is sort of spread out more diffusely, right, in a broader area. Really important in vision because it allows us to sort of create contrast in our vision, where, like, think about the lines, right, where you see a demarcation between light and dark. Really important to have lateral inhibition to do that, to sort of not have a blurring where we have distinct lines in our visual view. And a lot of that is generated through a neural pool called pattern called lateral inhibition, right? So again, lateral inhibition is allowing stimulus, right, to be received at a particular location, but inhibiting the surrounding cells to sort of make this guy stand out even more than it would otherwise. Okay.